Thank you so much, Uday, for that introduction. And it's just my request to two of our panelists to just share the mic. My apologies for that, but it's a large panel, and the topic, of course, deservedly requires a panel of this size as well. It's a panel that is very, very close to me, simply because I am a cancer survivor, as I was sharing with all of our panelists earlier as well. I completed 5 years in remission this year, in fact, on the 7th of March. So I'm a living and breathing example of someone who can fight cancer. And there have been several cases of cancer within my family as well. In fact, 4 members in a single family. So I understand the impact that cancer has on a person, that cancer has on people, that cancer has on families. And it is thanks to all our doctors and researchers here that we are all sitting alive, breathing. So can we have, once again, firstly, a huge round of applause for all the work that they do to ensure that the rest of us get to live. Thank you so much for that. Let me begin with, Dr. Sharma just gave us a very important presentation on some of the cutting edge work that is being done. But I want to begin with Dr. Srivastav. You've spent decades at AIIMS, sir. What is the single biggest shift you've seen in how we treat breast cancer in India today compared to 10 years ago?
Thank you, Devika, and good evening, ladies and gentlemen. Yes, we have observed a paradigm shift in the care of our mothers and sisters inflicted with breast carcinoma. Now we consider systemic therapy targeted to specific molecular signatures on cancer cells, along with immunotherapy and anti-cancer drugs as the mainstay of treatment initially. This results in complete resolution of tumor in about a third of ladies, enabling us to preserve the breast. And the other remarkable paradigm shift is that newer version of PET-CT scan for the metastatic workup to know where the tumor has spread with specific isotopes targeted to receptors of estrogen and HER enable us to facilitate a specific treatment to these metastatic foci in vital organs like brain, lungs, liver, and bones. So these are two important paradigm shifts that we observed in last decade.
Thank you so much for that explanation, Doctor. And I want to bring in Dr. Geeta at this point in time, whose research is what has enabled what Dr. Sharma was just presenting to our audiences as well. Dr. Geeta, before I ask you a little bit of a more specific question about Nirama and the work that you're doing, of course, the background of it was the fact that as far as certain cancers are concerned, especially breast cancer, which is one of the most prevalent forms of cancer amongst women, traditional screening can be a little bit intimidating and also inaccessible for many women. How has your approach and your breakthrough, powered by AI, now enabling more and more women to get screened? And if you can just share some of the data with us in terms of how many tests you've done and how many results have come out positive from that data.
Yeah, thank you very much for having me on the stage, and super thanks to Dr. Aishwarya ma'am and Kartikeya sir for running that Namo Shakti Rath. It's helping millions of women, starting with thousands, but I'm sure we'll reach millions very shortly. And it's an amazing, socially concerned, technologically oriented, and really impactful, drive that you're all doing. Congratulations on that as well. So coming to what are the specific gaps and how do we sort of address? What I have seen in the last few years of research in breast cancer screening, first and foremost, the statistics is alarming. Breast cancer is the top cancer killer in 159 countries, and in the rest is the second. That's it. So every day we are losing 2,000 lives globally. In India, it's 276 lives every day going, And it's one of the most curable of all the cancers. We have the specialist here, but it's one of the most curable of all the cancers because it is a soft tissue cancer, right? And you could just remove it, for example, remove the breast, and if detected early, that's the question. That's the key thing, right? If it is detected in stage 0, stage 1, you can have 95% survival beyond 5 years. Whereas if it is detected stage 3, stage 4, it can be as low as 10% survival beyond 2 years. This is the statistics. So we wanted to try and see how can we enable women to detect early, and what are the problems? Why are they not detecting? And there is a recent study by Dr. Budweiser, who is a very famous oncologist from TMH, ex-director as well. 1.3% of women in India, only 1.3% of women do a mammographic screening. 98.7% are mostly detecting the problem with hand. This is a technology era, and this is not what we can see. So coming back to specific things, so there are 4 A's that are needed to make breast cancer screening really adaptable in people. Affordability, accessibility, accuracy, obviously it has to be accurate, and last but not the least, acceptability. This is my own 4 A's, okay? So we try to address this at Niramai by using completely a different method of screening, which is primarily enabled with AI. So we use infrared imaging device to measure the temperature variations on the chest and convert the infrared map or temperature map into a cancer health report using 25 different machine learning algorithms working in conjunction. Of course, use of infrared imaging is not my idea. It's been around for some time, but it was not used because it's extremely hard to interpret an image which is all red and yellow. So how do we use AI to extract biomarkers which are new and convert that into a breast health report? So quickly coming to the 4 A's, affordability. You're able to now do a screening test with 10% of the cost. The equipment cost is less than 10%. The operating expense that's needed is also 10 to 15%. So we eliminate the cost of screening, and hence, of course, we can transfer that to the end users. Second, affordability, accessibility. It is extremely portable. We can hold the test, put it in a backpack, take it out and do the screening. We are doing the screening in Meghalaya, hilly areas, Uttarakhand, right, which is a lot of, so you can actually take it out and do the screening in corners of the world because it's just portable. It is radiation free, so you don't need a lead protection, walls and so on to do the infrastructure. You can just put a curtain, put it inside, and the lady can actually start do the screening. Accuracy, we have done several clinical studies today, 56 publications, where we have shown that we have high levels of sensitivity and high levels of negative predictive value, which means that when we say no, it's pretty much a no, but when we say yes, it means that she needs to go for further diagnostic workup. It's meant for a screening test. And finally, acceptability. People don't want to remove clothes in front of others, don't want to be touched, don't want to have those pain, because once they do the test, they run away. So we made it completely privacy aware, where we keep the device in front and click, 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 just like these cameras, we take thermal images instead of visual images, and that's about it. Before that, we blow cool air. And this is done in complete privacy in a full curtain like a changing room. So no one sees or touches the person during the test, and no compression, no test. So by attacking all the 4 A's, I think we've made a lot of difference to many women, and close to 5 lakh women have already benefited from this test.
That's such an, I mean, it's such an important breakthrough in terms of just accessibility, I would say, because there's such a stigma around certain, especially around breast cancer. But Dr. Geeta, I also want to understand, one is detection, the first step is detection, and the second is treatment. So talking about that a little bit, does the thermal imaging then ultimately help a surgeon like Dr. Srivastav plan a surgery better, maybe go for a lesser invasive procedure? Is that the future here?
Yeah, so thermalytics is a screening test, right? To screen, let's say, 100 people and say, who are those 4 or 5 people who need to go for the next level testing, which is more diagnostic imaging, where a mammogram or an ultrasound or an MRI can be done, effectively. There we actually even enable let's say an ultrasound, which is a radiation-free test, can be used for screening, but it's extremely subjective. So, but thermalytics basically marks the area of likely malignancy, or left breast, upper outer quadrant, 10 o'clock position. So with that, now ultrasound can be focused on that. And for the treatment, we are just looking at a few use cases, which is primarily when a person has a big lesion found. Instead of directly going to surgery, typically there is something called neoadjuvant chemotherapy, and Sir can explain more, where chemotherapy is done before the surgery is done. Right, and so that we can reduce the size of the lesion itself,
Yes.
and the surgery itself will be smaller, and less of the breast is actually affected. And so it is more acceptable for the lady. And here, what we have just started is a clinical study on it right now. The technique is there, where it's called longitudinal check monitoring. So can we figure out how the lesion is progressing or regressing? That is it becoming bigger or smaller over time? Just like you monitor sugar levels with a glucometer, can we use this thermalytics for monitoring the breast health? So with that, we know when possibly to stop the chemo, so that you can immediately know, like, there's no need to add more to this. So this is one. And a couple of other things like vascularity marking, so that we know where to sort of clip the key blood vessels which are feeding the lesion and so on.
Yes.
So these are potentially new use cases, because it's yet not fully clinically proven, but these are some exciting next steps that we are looking at.
Well, that's remarkable work. And with that, I want to also bring in our other panelists who are also doing cutting edge work as far as finding cures is concerned. All very esteemed professors with us, whose research then enables people like Dr. Geeta to take that forward into finding a cure and finding other things. And then, of course, the next level is Dr. Srivastav, who goes in with all the medical expertise. Dr. Tayali, I want to bring your work into focus here, ma'am. Your work, as per our research, focuses on hot and cold tumors. Is there a way that we can trick now, or through your research, have you been able to find a way that we can trick the immune system into seeing and attacking a cancer that it previously ignored?
Thank you, Devika, for this question, and also for setting the stage right for me to be able to answer the question through answers from Dr. Srivastav and Dr. Geeta. So as they've mentioned, the typical treatments of cancer have been either chemotherapy or radiation or surgery, or combinations of those depending upon the stage and type of cancer, right? But as many of us know, typically after surgery, if we go through chemo or chemotherapy, chemotherapy typically kills the cells indiscriminately, where because of that, we see side effects of cancer, such as losing hair and having nausea, vomiting, and so on. But of late, what we have is immunotherapy. So immunotherapy, which actually can, so if we know that cancer in general, cancer cells are the type of cells that actually somehow are able to evade the immune system, right? So immunotherapy is a type of therapy that is able to teach the immune cells to do what it is supposed to do, or basically to kill the cancer cells. So the hot and cold cancer types are basically those cancer types, typically solid tumor cancer types, where they are either infiltrated with immune cells or not so infiltrated with immune cells, which means they have a natural propensity for the immune cells to come in or not. Now, immunotherapy can actually help treat those tumors by actually specifically changing those immune cells, or teaching those immune cells to be able to treat cancer. And you may or may not have heard about the latest CAR T-cell therapy. So within immunotherapy also, there are multiple ways in which you can teach the immune cells. You can either have like an antibody-based immunotherapy, where the cancer cells are sort of tagged with a particular antibody, or the immune cells are tagged with a particular immune antibody, and so on, such that the immune cells are able to recognize cancer and kill. The most latest one has been the CAR T-cell therapy, which is a cellular-based immunotherapy, or an adoptive cell therapy, wherein the cells are taken out of the body, they are manipulated ex vivo or genetically engineered ex vivo outside of the body and then reinfused into the body. So this involves a lot of regulatory or operational costs, and this therapy has been very successful for hematological malignancies such as acute lymphoblastic leukemia, non-Hodgkin's lymphoma, and so on. So all the liquid cancers or blood cancers have been very well treated. Not all, but some of them have been very well treated with this therapy. But for many solid cancers, that's not the case. What we are doing in our lab is basically doing an alternative of CAR T-cell therapy where we can actually use a material-based approach, like a hydrogel-based approach, an injectable hydrogel, or an implantable scaffold material-based approach, where we can actually implant these materials and teach the immune system to, sort of get manipulated, and then, recognize the tumor cells on their own. So we are working on, sort of an alternative to CAR T-cell therapy, which will be useful for both solid tumors as well as, liquid tumors.
You just spoke about injectable hydrogels, ma'am. So on that note, I want to bring in Professor Das as well, because that's his area of expertise as well. Professor Das, as someone who's been through a very tedious chemotherapy regimen myself, and has first-hand experience of what undergoing chemotherapy feels like, I know that your work and your research will be really a game changer for the future, but how close are we, sir, to a future where chemotherapy only hits the tumor and leaves the rest of the body untouched?
Okay, so every time I see a cancer survivor, we feel that, or I feel that we are doing good. So congratulations on that, and thanks for having me on this stage. To answer your question, let me explain briefly what we are doing. We are trying to avoid the side effects of the chemotherapeutic drugs. So we all know that what is cancer in a very layman's language is an unwanted and uncontrolled growth of cells. So whatever drug we put in our body are basically poisons, killing the cells, and that's exactly why you see so much side effects. You lose your hair, you must have experienced all these things. So what we thought that for certain types of cancers, more which are tumor-based cancers, can we localize, deliver the drug localized way? But to deliver that, you need a vehicle. It should stay there for a long time. So we put it in a hydrogel. Luckily, we got a very interesting hydrogel, very small peptide-based hydrogel that we have made. And we incorporated a drug in that. It's an injectable gel, so we directly inject it on top of the tumor, and it stays there. This is the best part. It stays and breaks only in presence of glutathione, which is overexpressed near the tumor. So as the glutathione is expressed, the gel is breaking, becoming sol, and releasing the drug. So far, we got that after 18 days of single injection, we got around 75% reduction in the size of the tumor.
Wow, okay.
Now, you may say, why not 100%? It's a very time-consuming experiments, and we did all these studies during the COVID, so it took us almost 5 years to complete. My understanding that even after 18 days, when I see the gel remaining near the tumor, so within 30 or 36 days, maybe it would have been gone completely. But the beauty of this is that even if it is reducing 25%, what ma'am was telling, sir was telling, the surgical process will be very easy for many cases. Some tumors simply cannot be removed because of the way they have grown. So if we can reduce it by 25% or 30%, then the openings will come. Now coming to your question, yes, it will come. I'm very hopeful that in the near future, something like this will come up. The only issue is the very expensive clinical trial. That's a major challenge to break, and especially talking about India, Indian pharma companies are mostly concentrated towards generic medicine. They are not really interested in investing on new molecules. That's a big challenge that we have to overcome, but I'm very, very hopeful that we will be able to do that.
Well, that's absolutely amazing, body of work that everyone here is doing. Professor Biswal, bringing you into the conversations, I want to just understand from you that while, yes, several of our researchers are finding some of the newer ways to introduce drugs to reduce the impact, your work focuses a lot on drug resistance as well, which is a very heartbreaking reality for many patients today. How is your research into exomes now helping us predict and in fact entirely stop a tumor from becoming, one can say, immune to treatment?
Thank you for the questions, and thanks Kartikeya Sharma-ji and his team for setting this wonderful platform. And regarding your questions, currently for cancer treatment, a lot of therapies are available, and patients also respond very nicely to these current available therapies. But what happens after a while, means these therapies do not work, means these cancer cells become resistant to this treatment. Okay. So that's a big challenge for these clinicians to overcome this chemo resistance. Okay. So recently we found that in these tumor cells, they secrete small extracellular vesicles. They are called exosomes, which acts as a microscopic messengers. Okay. So they carry specific biomolecules like some protein signatures and some genetic variables, so which could act as a molecular signatures to predict when and how these type of cancers, whether these are sensitive cancers or these are chemo-resistance. So these will help the clinicians to set up the clinical status, which type of treatment it should be given. Next is these exosomes not only used for the predictions purpose, they can be used as a delivery vehicles. Okay, so we can use this current available therapy, and what we can do is a next generation therapy is not a monotherapy, it is a combination therapy. Okay, so we can combine two drugs together. Okay, in currently we are using phytocompounds also as alternative therapies. So with the chemotherapy, and we combine with the phytocompounds and encapsulated into these small vesicles, exosomes, then deliver specifically to the patient. Okay, and what Mama told that the immune cells, okay, so we can also derive these exosomes from these immune cells, that is called immune cells derived exosomes, and these exosomes can be used to train other type of cells to fight against this cancer. So in that way, these exosomes could act as a next generation molecules to fight against the chemo resistance type of cancers.
Okay, all right. Again, once again, thank you so much for your work. And I know the amount of effort it takes to be a researcher, a doctor, and Dr. Srivastav, you've had such a glorious career, sir, and thank you so much for your service to humanity via AIMS as well. Dr. Savan, I come to you. So we've spoken about detection, about treatment, about drug resistance, but looking at your body of research and work with precursor lesions, is the ultimate cure for cancer going to be the ability to finding it before it becomes a malignant tumor?
Thank you for the question, Devika ji, and thank you for the honor, Dr. Pandit Sharma and Mr. Sharma ji, and thank you for this opportunity to share the stage with such august scientists. So as a physician who now works in clinical trials, let me first give you a bird's eye view of what precursor lesions are. I had the opportunity to work in the field of pancreatic cancer, screening subjects who had an IPMN. An IPMN is an intraductal papillary mucinous neoplasm, which is more often than not an accidental finding, which means you don't have any signs or symptoms and you go in for a regular screening or other GI problems and you accidentally happen to find an IPMN, which is again the biggest unknown or the dark side of pancreatic cancer, is that we find it at a very late stage. So it is correct that an early detection is going to be our ray of hope when it comes to such cancers. And the tripod on which the regime of pancreatic cancer currently is based on is screening, monitoring, and follow-up via monitoring. So these three things are critically important, and we are still working a lot, and like I said, there is so much unknown about this disease than there is known. I'm hoping that AI, with the help of gene sequencing and vaccinations that are in the pipeline for other cancers, lead the way to pancreatic cancer and help us find a cure. And I hope that in the next few years, very soon, I can sit here with you and discuss the cure for pancreatic cancer.
I hope so as well, as always. And Dr. Srivastava, before the conversation started, you were telling me that we don't use the word cure in medical terms, but hopefully there'll be a future, sir, where the word cure will be medically accepted as well for cancers. I've run out of time, but I'll take the liberty of just extending this panel by just a few minutes, because I want to pose one last question to the entire panel. Vaishnavi, we'll start with you. If there's one common myth about cancer that still persists in the Indian public's mind, or generally in the public's mind, what would it be and how does one dispel it?
I think the biggest fear about cancer is that when you get diagnosed, you're counting your days. And I think that is the inbuilt fear in humankind that we need to change, that there is a lot of advancements that we've made and you haven't lost the battle. You are a fighter and you will be fighting it. It's going to take some resilience. It's going to take help. And like Dr. Pandit Sharma said, we have to build a community. But the biggest myth is that that is the end of your life. And we together sitting on this panel, along with everyone else, with a public-private partnership and the government of India doing such wonderful work, is going to fight it together.
Professor Biswal, same question to you, sir.
Yeah, so for this one, what happened is first we have to remove the fear among the public. Okay, so that's the first things. Second is what ma'am is doing, like screening. Okay, and screening, if we take the invasive, that so again fear will not go. So if we can do like a non-invasive screening, like ma'am is like thermal screenings, or like extracellular body fluid, what we are now targeting the saliva or like urine. Okay, so that type of non-invasive screenings will help for early predictions, and fear will automatically go. And if early predictions is there, treatment will definitely be much better.
Right, absolutely. Professor Tyaria, just 30 seconds, ma'am, one common myth.
So the one myth, again, one is that there is a cure for cancer. It's not like completely incurable, at least for some of them. However, the thing that people have to know that there is no one thing treats all kind of a thing. It has to be much more personalized for each person, each individual, the type of cancer, the stage of cancer. So the treatment has to be different for different people.
All right. Professor Das.
So the myth is, if detected, I'll die, and we are trying to break it. That's it.
Absolutely. Dr. Geeta.
While I agree with all the comments made, as an AI scientist, I want to say in this context, AI will not replace doctors, but doctors not using AI will get replaced by doctors using AI. So we need to collaborate together, and we can definitely fight cancer.
That's a great way of putting it, ma'am. Dr. Srivastava is the senior most person on our panel. I'll leave the last word to you, sir.
Of Asian patients in India, in Nepal, in Bangladesh, they think that biopsy and cutting the tumor will cause a spread of the tumor. And without biopsy, we cannot initiate any treatment. So that's the dilemma we face. So that's a myth that we have to dispel.
Absolutely. On that
Thank
note,
you.
a huge round of applause for all of our researchers, our doctors, and everyone who's doing cutting-edge work in curing cancer.
Thank you all. Thank you. What an inspirational panel that really was. And such change that is happening really also makes, I'm sure, our audience feel very motivated as well to do their bit. Okay, well, we are shifting our focus back once again to AI. And in the segment of Next in AI, we are going to now turn for another keynote address to a very respected voice. An academic, someone who, of course, has big ideas himself, but is going to be speaking to us about the big idea, imagining a digital economy for all. I welcome on stage Dean of Global Business at the Fletcher School, Tufts University, Professor Bhaskar Chakrabarti. Could we have you on stage, please? Let's keep the applause going for Professor Chakrabarti.